Hello, everyone. My name is Monika Lipińska, and I will be presenting the BIO-TRLs project, which is ongoing work. So this is not a finished product or finished based on finished paper. This is something that, together with my colleague Layla Van Ellen, who's actually leading the project development, we're working on right now in order to develop this evolved technology readiness level for biologically active materials and organisms. The context is that there is a lot of ongoing research and developments on biological materials and organisms and living materials, and in general for the use of biology for space applications. The examples of that: plants for life support systems, microorganisms for recycling, extramorphiles. I'm personally working with mycelium for construction materials. There's research on melanin-rich materials, microorganisms for metal extraction and processing in soils. So there's a lot of research ongoing for how we can use different biological organisms, microorganisms for different applications in space applications. So it's not like I'm working, for example, with the scale for construction, but the developments and the ideas for different projects and applications are on a variety of different scales. You can change slide. And yeah, didn't change. Okay, thanks. And to assess technology readiness level, we have Technology Readiness Level scale that was developed by John Mankins, working for NASA. And that scale now is used for a variety of different things. It's not specifically used for space applications, but it originated from space applications. So it's basically how to assess the readiness of the technology, starting from TRL1, which is basic principles observed. And then between 1 and 4, it's the research lab. So the early development of the project with laboratory testing and development of the prototypes. Then 5 and 6, it's simulations. So it's usually simulations in the responding environment. So specifically for space application, that would be in as close as possible to space environment. And then finally preparing it for the flight. So integrated pilot system demonstrations and actual system ready for full-scale deployment, which is TRL9. So TRL9 now for different applications we kind of call like an off-shelf product. So it's something that you can go to the store and just buy it. That's TRL9. So it's completely finished product. But it's much more challenging when working with biological materials. Victoria, can change slide. So the original technology readiness level system was developed keeping in mind that the technology is developed on Earth, tested in analog environment, and then it's shipped to space. But the product that we're developing, the technology that we're developing, it's not really changing. And then if you can click. But then when we're working with biological materials, especially now when we're, the idea behind using biological material, one of the main ideas is to save up mass volume. So we're using biology so we can grow things in situ directly at destination. We can manufacture drugs or different products, and in my case, construction materials, for example, in situ. So we're not having final product that is fully developed on Earth, tested, and then we ship it. It's something that needs to be developed directly at destination. And that's where the main challenge comes, because we cannot really simulate the full environment, how different it will be on Moon or Mars or wherever we want to, whatever we want to target. And that's the main challenge with using biologically active materials and using technology readiness level scale. But also we want to assess the readiness of the technology or biotechnology that we're developing. So that was kind of the problem that we stumbled upon during our research on biological materials for architecture and for space architecture. And based on that, we decided to look into how to develop BIO-TRL level scale that would respond to all these challenges. If you can click further. So yeah, so as I said, the environment is the main challenge. So the behavior of living materials or organisms cannot be accurately reproduced in analog environments. Then the next challenge is the scalar matters. So if you can click further, scalar matters, it's kind of two different challenges in one, where working with biology, even if you prove something, you have a proof of concept on a small scale, you start scaling up, that the whole system, the whole biological organism, the technology that you're developing starts acting differently. It's not the same. It doesn't scale up linearly. So that's another challenge. Like we cannot really say that we have a proof of concept, let's scale up and that's it. We have to figure out how to assess the technology readiness level on each scale throughout the development of the technology. And then the other challenge is technology assessment for microscale and nanoscale products. It's hard to call it a product, but basically there are certain developments that cannot be this TRL9 by itself because they're just a part of the bigger system. So they're components. So they're fully developed. They're developed as much as they can be, let's say, but there still wouldn't be, or maybe they would, TRL9. So it's hard with using the current scale that we have. It's hard to assess these technologies that are part of the bigger system. And then the next slide, I think it's also talking about a system and subsystem interactions. So we can have, this is, the image shows here my PhD research where I'm developing bricks using mycelium. So the mycelium is used to create one brick, but also there's interaction between multiple bricks, how they're growing together to create the actual habitat structure. So this is a very simple example of you might have figured out something on a scale of subsystem or level of subsystem, but then it's completely different when it comes to the whole system. So there's a lot of different challenges coming with working with biology. And the next slide, please. So in order to understand how to approach the development of the new technology readiness level, we analyzed different technology readiness levels, or not necessarily technology, because based on the original TRL, there are new systems developed like manufacturing readiness level, integration, innovation readiness level, and new fields are taking this system and adapting to their field. So adapting it to biology, it's not like completely new idea. Different industries and fields are already adapting the system, but we feel like the biological one really needs like reorganization. Also, when it comes to dimensions, most of these readiness levels are having just one dimension. So it's just from 1 to 9, usually to 9, sometimes to 10, sometimes to 7, but there's no other dimension. But then having all of the challenges that we encounter, like working with different scale or subsystem and system, we feel that we need more dimensions to it to be able to really assess the technology and to understand the development level of the project or the product. If you can change slide. So we, 2 years ago, we organized a symposium on biofutures for transplanetary habitats, where a lot of projects were presented, and we took these projects as our case study for like to start thinking of, okay, what should we think about while developing the new system? And we mapped them on this biogram diagram to understand which fields they're covering between like biomaterials, bioengineering, and biotechnology. Most of them are somewhere around biotechnology. So that's probably, that will be probably the field that we will focus the most for now, because we see that like the spectrum, the scope of the projects that are connected to space applications and biology are mostly around biotechnology. So this was just to like understand our scope and like have the starting point. If you can change slide. And this is also an example of how we look at different scales. So we mapped these projects also on the different scales. And then if you can change slide, there will be example of one, I think the bioreactor, where it should, Victoria, can you change slide? Okay, exactly. So the bioreactor for Mars, for example, it's like marked on the micro scale, but actually like there is this line going all the way to like scale of millimeters, meters, because the idea is that the actual final product would be on the big scale, but right now developments are on this very micro scale. And then if you change slide, there will be another example of Melissa project, which is simultaneously developed on different scales at the same time. So this is also another challenge, like how we can reflect that while assessing the technology. Where are we with the technology? Okay, and then if you can change slide again, and like here you can see Melissa like on different scales at the same time. And then the next one. Okay, so basically this project, it's not something that, as I said, it's not something that I'm presenting you what we came up with. It's more like, this is what we want to do, and we have certain approach to that, but we really seek feedback and input if anyone has any ideas of how we can take it further. Our next steps are actually, now that I see the slide, it's not next steps. This is like a summary. That it's critical to have a differentiation between system and subsystem, as I said. The differentiation between analog environments and real environments should be further defined and developed to account for interactions between biological systems. Because, like, for example, contamination, like it's very challenging to work with biological systems. So the environment itself, the environment that we're targeting itself, plays a huge role in the development of the product. We cannot just say it's finished and take it wherever you want. It's really interconnected with the whole development of the system and how to grow it in situ and produce it in situ. And to gather this data in more depth, we're planning interviews with different experts and stakeholders. So this is where, yeah, what I was saying, like we're looking for collaborators and for people who are willing to like share their experience with working with biological materials and talk with us, and maybe together we can think of a way of how to develop this system. If you can click further, I have a few more like, yeah, so basically we started developing this interview canvas to see like what information we want to get to start with creating the new system. We started with case study based on my PhD project, the mycelium components, where I tried to look at my project having this lens of, okay, let's try to think of it as as subsystem interactions and system interactions, and like from the perspective of technology development, what I can learn from this project to guide us for development of the technology readiness level. So if you can change slide. So we developed this interview canvas, and now it will be a couple of slides just like highlighting each part. So maybe we can just skip that. Maybe we can stay on this one. But basically, this is like a very beginning where we're thinking like we have this mirror board and we want to invite people and to just together with them basically go and fill in the canvas where we want to have like this general description of the project, but then also mark the stage. So like, first of all, what would be the aim of the project? And then what procedures are involved, like simulation, experiments, analogues, other, is it system or subsystem? And then we have like suggested different development stages. So this is kind of based on the regular first TRL technology readiness level, but we didn't want to say the numbers because the whole point is that we don't ask you to assess your technology based on what is existing. We're trying to figure out how to navigate with the new system. So like it's more descriptive and qualitative assessment to understand like how do you feel where your technology is, and then we want to assess also what is the scale. And then that's one of the ideas that we can go through many different projects and then find a way of analyzing the data. And then the other thing is, if you change slides, yeah, you can just click through these because I just said it. And then, yeah, and then the other thing is that we want to create this ecosystem maps and metabolism maps to understand the interactions of specific technologies, products, materials. So this is again based on example of my projects where I was looking up the interactions between different stages of my project, between fabrication stage, assembly stage, and the growth stage, and what are the factors affecting the growth and the production of my samples. So like on this overall level, let's say the things that are affecting the growth, it's temperature, humidity, time, the blend of what I have. And then like we kind of go deeper that mycelium types and the geometry are affecting the assembly process. And then finally the mold is affecting the fabrication. So this is again simpler version. I assume that if we go to like more complex biological systems, this ecosystem maps would be much more complex. But it was just to like picture what we have in mind. And then if you change slide, Victoria, there will be also metabolism maps. So this is to understand the components that are playing important role in the creation of the product. So what exactly are you using? Because again, each component is playing a big part in the creation of, like in my case, in the creation of the brick. So we want to understand how many variables they're coming into play. So this is just step-by-step processes. And we also prepared that more interactive. So we can invite people working with different biological products, and then they can kind of interact and create their metabolism maps. And then the next slide shows also more interactions. So it's not step-by-step, but it's interactions to understand how each component is interacting with other component. I'm not sure yet how we will analyze this data and how we will get the answers, but these are like the scope of things that we thought of that could be possible to analyze and could be comparable, could be made comparable between projects. So I don't have really any real outcomes. This is all discussion, and I invite you to reach out to us. Yeah, you can totally change slide now. I invite you to collaborate with us, give us feedback. And if any of you is working with any biological living materials, I would greatly appreciate if you contact us. And then if you will be willing to participate in one of these interviews where we just go through your project and we try to understand the interactions and development process. Thank you.